Okay, I'm sure you guys were waiting with bated breath for part 2 of Lecture 7, so let's get going. I know I left you with a cliffhanger. So, backing it up a little, there's two main divisions of the cell cycle: interphase, when the cell is not dividing and just doing cool cell things, and the M phase, where the cell is dividing. Well, when we think about interphase, there's three events that occur, three intervals of time that occur that I want you to know. But this is still when it is not actively dividing. G1 is the stage before before DNA replication. This means this is where we're getting ready for other things. The cell doubles its organelles, so it's making a lot of membranes for new Golgi, new rough ER, smooth ER, etc., etc. It gets everything prepared for DNA synthesis, which is where DNA is going to replicate. And there's actually a checkpoint to make sure everything is prepared to go through the rest of the process. And if it's not, it goes into kind of a stage of pause, where it stops going forward in the cell cycle. It's arrested. We call it G0, that little circle, we call it G0. When it's not going to divide, that's not really what it means, but you can memorize it that way. And so this makes sense for cells that don't divide, like our nerve cells and our muscle cells. They do go through part of the cell cycle where they grow to a certain amount. They actually can go through other types of growth where more organelles are made in certain parts, but yet then they're halted. They never go through of the actual division to become more cells. The second part of interphase is the S phase. S stands for synthesis. So this is where DNA synthesis takes place. And we're not going to go into the nitty-gritty details of DNA replication, but essentially we're copying the DNA so that we have two sets. So at the beginning, it's in chromatin form still, because all of interphase DNA is in chromatin form. But now we're going to copy it, and we're going to put those two copies together at the centromere, and then we will start the process of condensing down the DNA into chromosome form. Okay? G2 comes after that, and that's right after DNA synthesis. And this, again, goes through some checkpoints to make sure we have all the needed chemicals to go through cell division. So again, thinking about this picture, the blue this time is interphase. So we have G1. The G stands for gap. If it's not going to divide, it kind of stays here in G0. If everything's good to go, to proceed with division, we need to copy the DNA. That's the S phase. And after the DNA is copied, we're going to do some more checks. We're going to make sure the cell's big enough, and that happens in the G2 phase. All of that is interphase, but it can be separated into three parts: G1, S, G2. The M phase occurs after interphase if everything's good to go. The M phase can be broken down into two parts. We have mitosis, which is the division of the nucleus. So that's where the actual nuclei, you go from one nucleus to two nuclei. And all the DNA is in there, of course, because that's what a nucleus is. And then the second part of cell division, the second part of the M phase, is cytokinesis, which is the division of everything else. So the cytoplasm and all the organelles, and at the end, you get two identical daughter cells. So if you look, you might remember the words of prophase, metaphase, anaphase, telophase. We are going to quickly go through them. You're not going to see pictures on a test where you have to differentiate the phases. I think knowing them in order and knowing a basic event of each one is important, so I'm going to ask you to know that. But again, this is still the same cell cycle. It's just we can break it up into two parts, interphase and M phase. We can break up interphase into three parts, G1, S, G2, and we can break up M phase into mitosis and cytokinesis, and then we can further separate mitosis into prophase, metaphase, anaphase, and telophase. So this is a familiar diagram, right? So we have our DNA replicated. That happens in the S phase of interphase. And now we're getting ready to go through mitosis. And at the end, each daughter cell gets one of each of these chromosomes. Something else to think about are these things called spindle fibers. Okay, so spindle fibers are throughout the cell and are part of the cytoskeleton. So again, we've already talked about cytoskeleton. It keeps the cell in a certain shape, but they become activated during cell division to help separate out the DNA. Okay, because again, humans have 46 chromosomes, 46 little X's in there, and we have to make sure that each daughter cell gets one when we separate those X's out. And we also talked about an organelle very briefly in the cell lecture where we talked about that centrioles help cell division. This is what they do. They direct the spindle fibers. They organize them to make sure that they're lining up the way they should so that they can separate out the chromosomes. So when we talk about mitosis, and believe me, these actually can be separated into more than 4 phases, but this is what you learn in high school, and we'll learn it again now. There's prophase, metaphase, anaphase, telophase. I tell my students to remember PMAT, like you're commanding Matthew to go to the bathroom. Hopefully, like actually in a toilet and not in the corner of your dorm room or something like that. That'd be really gross, huh? So P stands for prophase. And this is where the chromosomes are visible. They look like little X's under the microscope. They are in condensed pairs. See what I did there? P for prophase, P for pairs. The second phase of mitosis is metaphase. Metaphase means middle. Meta does. And this is where the chromosomes line up in the middle. Then comes anaphase. This is when the chromosomes are pulled apart to opposite sides of the pole. And then we have telophase, and this is where 2 distinct cells and 2 distinct nuclei are visible under the microscope. They pack in cytokinesis, which actually is not part of mitosis officially, but it is part of M phase, and that's when we're going to separate the cytoplasm and everything else. So I've got some pictures. So up here at the top, these blue things are actual microscope pictures of cells going through mitosis. These look like cheek cells to me. There's probably some kind of epithelial cells. So, and then down below is kind of the cartoon version, right? So here's a normal cell. We've got the nucleus. It's got some pores, right? We know that the nuclear envelope has pores in it. And inside is chromatin, which is DNA, but it's in a nice relaxed form, really thin. Once we start, and that's all of interphase. So that's G1, that's G2, G1, S, and G2. If I say them in order. Then when we get to M phase, we're going to start the phases of mitosis. So here's prophase. Prophase is actually pretty busy, but they are seen in pairs, and the nucleus starts breaking down. By the end of prophase, the nucleus is gone, and here are little centrioles here. They call them centrosomes here. Pretty much the same thing. So the centrioles start organizing the spindle fibers between them. And that's prophase. Metaphase, the chromosomes are going to line up in the middle, and the reason they're lined up that way is because the spindle fiber is attached to that centromere, that little belt around the 2 copies, and line them up in the middle. Sometimes we call it the equatorial plate, right? It's like the equator. Anaphase is when they're pulled apart, noting that each pair, the chromosome's been replicated, so each sister chromatid is going towards opposite ends of the cell. And then in telophase, and I forgot to say, you can be fancy and say telophase if you want. In telophase, we're going to start forming 2 nuclei, and we're going to start pinching in the cell membrane to start that separation. And you can see that even under the microscope here, which is pretty cool. So cytokinesis, for just a minute, is when the cytoplasm, the organelles other than the nucleus, and the cell membrane divide. And essentially what happens is it starts pinching in a place called the cleavage furrow. No giggling, please. And then we're going to keep pulling that apart almost like a string, right? Kind of like if you play with slime or something, right? And you have two handfuls of slime and you pull it until finally that last thread breaks and you have it separated. That's kind of what happens during cytokinesis. There's a type of cytoskeleton. It's the same protein that's in our muscles called actin that actually kind of constricts it so that they can separate out. That's in animal cells. Notice there's no cell wall. It looks a little different in plant cells because of the cell wall on the outside. In plant cells, we actually get this cell plate that forms. It goes across this way. This is the edge. And then in between the two new daughter cells, if we zoom in here, we would see these vesicles that contain phospholipids that start forming the boundary between the two cells. Then eventually new cell wall components are also formed there too. So it looks a little different because they don't have the opportunity to kind of pull apart. So instead the wall gets built in between. All right, I am going to pause here because I don't think I can get through the last part in one go. So this will be the end of the second part, and we'll get through the third part in just a minute.